We are going to demonstrate two protocols today that demonstrate how to study NK cell function. There are two key aspects of NK cell function, that is its ability to eradicate tumor cells and its ability to migrate to tumor microenvironment. These two protocols will demonstrate the NK cell ability to eradicate tumor cells and also its ability to migrate to tumor microenvironment. So the two protocols that we are going to demonstrate today are straightforward, does not require use of radioactivity, and can be established in most labs that has the ability to do molecular biology and cell culture work. Suresh Bugide and Suresh Chava, who are two postdoctoral associates in my lab, will demonstrate these two protocols today.
To assess NK cell-mediated cytotoxicity using LDH, first grow a human liver cancer cell line to 70 to 80% confluency in a 100 mm cell culture petri dish at 37 degrees Celsius and 5% carbon dioxide. On the day of the experiment, wash the culture with 5 milliliters of PBS before treating the cells with 1 milliliter of 0.25% trypsin EDTA until the cells have detached from the plate bottom. When a single cell suspension has been obtained, add 10 milliliters of cell culture medium to the cells and transfer the cells to a 15 milliliter conical tube for centrifugation. Wash the cell pellet with 5 milliliters of PBS before resuspending the cells at a 1 × 10⁵ cells per milliliter of fresh culture medium concentration. Next, seed 100 microliters of the target human liver cancer cells per well in triplicate per treatment condition in a 96-well plate, and add 1 × 10⁵ human NK cells in 100 microliters of